Hello and welcome to today's webinar hosted by Kapa Biosystems. My name is Deepthi Sanjay, and I'm the product manager for RNA sequencing, as well as moderator for this event. Before we begin, I'd briefly like to go over some GoToWebinar features to help ensure that everyone enjoys our presentation. All attendees have been muted for the call in order to provide the best sound quality. We will, however, be taking questions, so please feel free to type them in the questions panel throughout the presentation, and we'll attempt to answer the requests at the end of the webinar within our 1 hour allotted time frame. Today's presentation will be recorded and hosted on Kapa's website later this week, and an email notice will be sent to all attendees. For those of you not familiar with Kapa Biosystems, we are a global life science reagents company focused on evolving enzyme product solutions for various applications, including PCR, qPCR, and next-generation sequencing. Listed here are some of our core competencies, which include enzyme engineering, production of high-quality reagents, and scientific support. As previously mentioned, one of our strengths is high-quality enzyme engineering using our directed evolution technology. Directed evolution is like natural selection in a test tube. Essentially, we use a combination of random mutations and an artificial selection pressure to evolve enzymes with improved properties. In this example, we start with the gene for wild-type Taq polymerase. We then introduce millions of random variations by mutagenesis. We clone the gene library into an expression vector, transform it into E. coli, and express the mutant proteins. These proteins are compartmentalized, and the gene expression variants are expressed in emulsion. So this is similar to collapsing hundreds of 96-well PCR plates into a single tube. When this emulsion is made, we also add the selection pressure and use this to screen millions of variants. Only improved variants survive the selection pressure, so mutants with no advantage disappear from the gene pool. And this results in enzymes with performance improvements. We repeat the process multiple times with the surviving mutants so that we're left in the end with the best mutants. We then pick the best one for the final stages of product development. The great thing about molecular evolution is that we get extra benefits out of the process. For example, our enzymes are much faster, more robust, and better at amplifying both GC and AT-rich sequences, which we can take advantage of to produce high-quality kits. So we've used the advantages that we gain from our technology to address the needs and challenges of transcriptome sequencing. Some of the goals of transcriptome sequencing include discovery and annotation, quantification of gene expression levels, and determination of the transcriptional structures. It's a powerful tool for investigating RNA, but it still has some challenges. Here are some of the challenges of transcriptome sequencing. These include achieving sufficient coverage for GC-rich regions as well as low abundance transcripts. Ability to accurately assess transcript orientation as well as minimizing bias during library application are some of the other challenges associated with RNA sequencing. At Kapa, we manufactured our RNA-seq kits to address these challenges. With that, I'd like to introduce you to Dr. Adriana Geldart, who will discuss optimized library construction for high-quality transcription sequencing on the Illumina platform. Dr. Geldart has a bachelor's degree from Stonehill College, a PhD from UMass Medical School, and completed her postdoctoral fellowship at Brigham and Women's Hospital in conjunction with Harvard Medical School. at I'm Torrent. With that, I'll hand over the presentation to Dr. Geldart.
Thank you, DC, for the introduction. During the webinar, I wanted to discuss the following 3 topics: how we've developed appropriate QC methods for assessing the performance of RNA library construction workflows, the development and validation of an automated solution on the PerkinElmer Cyclone GFX Workstation, as well as some validation data generated with a key opinion leader in the field. Shown here is the Kapa-stranded mRNA-seq workflow. This workflow contains an mRNA capture step. However, this protocol can be used with other RNA as well. In short, an RNA sample is taken through mRNA capture with magnetic oligo-GP beads. Captured beads are then eluted. The captured mRNA is then eluted from the capture beads, followed by fragmentation of mRNA in the presence of magnesium under elevated temperatures. The first-strand cDNA synthesis is performed using random priming, followed by second-strand synthesis and marking. This step will convert the cDNA RNA hybrid to double-stranded cDNA and also incorporate the UTC into the second cDNA strand. The cDNA is then progressed through a pairing, adapter ligation, and amplification using Kappa Hi-Fi hot start DNA polymerase. Kappa Hi-Fi will stall when it encounters the uracil base that is incorporated into the second strand, thereby preventing amplification. Therefore, during PCR, the only strand that is fully and completely amplified is the first strand. Strandedness is then maintained by the directionality of the adapters, as the P7 adapter will be on the 3' end of the cDNA strand. For the Kappa-stranded mRNA-seq workflow, we have taken what we have learned from our DNA preparation methods and have applied it to RNA. Therefore, many of the key features that you see in our DNA library preparation kits are applied to this workflow as well. First, the fragmentation step, we have optimized conditions for various insert sizes. The first strand synthesis is performed by a modified MMLV reverse transcriptome called Kappa script. Moving on with the library preparation portion, we again make use of the with-bead protocol. This means that each of the enzymatic steps are performed in the presence of ampere beads. The benefits of a with-bead workflow include improved DNA recovery. It accommodates DNA inputs down to 100 nanograms or less. It reduces the cost of spry beads by 60% and also results in higher yields of adapter ligated library molecules. In addition, we have optimized the adapter insert molar ratio to allow for optimal formation of adapter ligated molecules. We have also optimized post-ligation cleanups to remove residual adapter and adapter dimer from the final library. And again, we implement the use of our Kappa Hi-Fi hot start DNA polymerase for low bias amplification. We have also worked towards developing key QC methods throughout the workflow that you can use to assess library quality. While it is very easy to make a library, what you ultimately would like is to make the best possible library from the samples that you have available. We have essentially broken up the workflow into 3 different modules and identified key metrics to assess each block. This will enable you to determine where an issue has arisen during library preparation if necessary. Also, identifying key metrics to assess library quality is important because it allows you to evaluate the library during a manual evaluation as well as during development of automation methods without actually having to sequence the library. The first module is used to evaluate mRNA capture and fragmentation. The first QC metric we have incorporated in this block involves determining both the concentration of the input RNA by a qubit assay and also examining the electrophoretic profile of the input RNA to assess the quality. Generally, for a poly-A capture, we like to see RNA integrity scores of 7 or greater as assessed by the bioanalyzer chips. After fragmentation and priming, we also like to look at the fragmented DNA, as there can be some variability between thermocyclers and other lab equipment such as tubes or plates. Prior to using the Kappa-stranded mRNA-seq kit, it is recommended to perform a small pilot study to optimize the fragmentation parameters according to your specific workflow. Also in this stage of the workflow, we examine 28S, which is an rRNA-specific gene, and GAPDH, which is a housekeeping gene. We examine their expression levels by using the Kappa one-step QRT PCR kit. 28S, again being an rRNA-specific gene, will be depleted from capture mRNA as compared to the total RNA, whereas we won't expect a difference between captured RNA and total RNA in the housekeeping gene. Please keep in mind that depending on what species you're working with, these genes may need to be optimized for your particular workflow. The second module starts from cDNA synthesis through adapter ligation. A metric we look at the end of adapter ligation will calculate the conversion rate to adapter ligated molecules. We do this by performing a Kappa library quantification kit analysis post-ligation cleanup. This is the most important metric to examine, as it does give key insight into the success of the workflow. As we are not quantifying cDNA material prior to A-tailing, we assume a 2% conversion of total input to cDNA for this calculation. Lastly, at the completion of the protocol, we examine the profile of the amplified library to observe the final library size and whether there are any adapter dimer present in the final library. At this point, we obtain an accurate quantification of the concentration of our libraries before proceeding into sequencing or a capture workflow by the Kappa library quantification kit. These next sets of slides will walk through each of these QC metrics and show typical results during library construction. Shown here is an electrophoretic trace of universal human reference RNA from Agilent. Agilent's universal human reference RNA is composed of total RNA from 10 human cell lines and is typically used as a control in our experiments. This particular sample has a Rin score of 8.2 and therefore is acceptable for continuation into the workflow. This parameter is important for mRNA capture, but may not be important for other methods such as ribodepletion. After fragmentation, we analyze the electrophoretic profile of fragmented DNA. As I mentioned, it is important and it is recommended that prior to moving into your library construction workflow, you perform a small pilot study to determine the optimal fragmentation parameters for your workflow. In this particular pilot experiment, we started with 500 nanograms of UHR RNA and proceeded through fragmentation under various conditions. Following Ampere cleanups, these samples were run on a bioanalyzer Pico 6000 RNA chip. The red trace shows the profile after shearing for 8 minutes at 94 degrees. With this incubation, we are obtaining a mode size slightly smaller than 200 base pairs. The blue trace shows the profile after shearing for 6 minutes at 94 degrees. This incubation step yields a mode size slightly larger than 200 base pairs. Lastly, the green trace shows the profile after shearing for 6 minutes at 85 degrees. This yields a slightly broader peak size with a mode at around 400 base pairs. The next QC step involves the use of the Kappa CyberFast one-step QRT PCR kit. This kit has been optimized for rapid one-step, one-tube RNA quantification. It allows us to take an RNA sample and perform both reverse transcriptase and qPCR in the same tube, whereas the two-step protocols involve performing the reverse transcriptase and the qPCR in 2 subsequent reactions. So the main features of using this kit is that it requires minimum hands-on time and thus reducing contamination risks. The PCR protocol is very fast. It exhibits high accuracy, reproducibility, and highly specific and consistent results across a wide range of input RNA and even with low quantities of target RNA. And lastly, speaking to the inclusion of the Kapa CyberFast enzyme specifically, it allows for earlier CT scores, ideal PCR efficiency, high fluorescence levels, and efficient amplification with a range of amplicon lengths and types. In this particular QC step, as I briefly mentioned earlier, we analyze expression levels of both 28S and GAPDH. The CQ scores of the captured mRNA is then compared to the CQ scores of the total RNA sample. Shown here are the results, again, when analyzing UHR RNA. As you can see, there is an increase in the CQ score with the captured RNA as compared to the total RNA sample, specifically for 28S, which is shown in pink. This indicates that there is a substantial decrease in residual rRNA that is measured post-capture. Typically, we see a change of about 8 to 12 cycles when examining the CQ scores of 28S. On the other hand, there is no loss of the highly expressed gene, and CQ scores are very similar between the total and the captured RNA for GAPDH. This indicates efficient capture of the mRNA. These trends are seen across all inputs that we analyze, 100 to 4 micrograms of input material. As a reference, I have also included the primer sequences for both GAPDH and 28S that were used for this particular QC method. Again, depending on what RNA you are examining, you might need to design new primers, and they should ideally be small amplicons, less than 75 base pairs. The next QC metric involves quantifying the amount of adapter ligated material with our Kapa library quantification kit. qPCR is widely regarded as the gold standard for NGS library quantification, as it accurately measures the number of molecules that can actually serve as templates during amplification. Compared to other methods, qPCR is the only method that quantifies only those molecules that are adapter ligated. To elaborate, following the ligation of adapters to library inserts, you're left with a mixed population of molecules, some with the correct adapter configuration and some without. The bioanalyzer, Pico Green, or NanoDrop will measure total nucleic acid concentrations, regardless of whether the molecules are properly adapted or not. On the other hand, qPCR will only measure those molecules that have adapters on both ends of the library fragments. In addition, because other assays employ double-stranded DNA binding dyes, the forked adapters, as they do contain a single-stranded component, will be under-quantified. Further, ligated material will run very strangely on a bioanalyzer trace, as the single-stranded ends will retard migration in the gel matrix. A metric that we will speak of often in regards to adapter ligation is the conversion rate, or the percent conversion. This is defined as the amount of input material going into library preparation, in this case the A-tailing reaction, that is converted into adapter ligated material. Because we do not specifically calculate the concentration of cDNA going into A-tailing, we assume a 2% conversion of input RNA into cDNA. The conversion rate can then be calculated by taking the amount of adapter ligated material, as quantified by the Kapa library quantification kit, divided by the amount of material going into A-tailing reaction. For the Kappa-stranded mRNA-seq kit, we typically see conversion rates of 5 to 20%, depending on the input amount and sample type. Shown here are the conversion rates using our UHR sample again. We observe a successful conversion to adapter ligated molecules falling in the range of 10 to 20%. As expected, the conversion rate also increases as the amount of RNA input is increased. Performing a library quantification kit post-ligation is also an invaluable tool in preventing overamplification of libraries. As you are determining accurately the amount of adapter ligated molecules, you can then empirically determine how many cycles you need to obtain enough material for QC, archiving, sequencing, or capture-based workflows. We also implement using the library quantification kit to examine final library concentrations. As mentioned, optimal cluster density is dependent on the appropriate concentration of PCR amplifiable DNA molecules, and therefore qPCR is the most accurate method to determine library concentrations prior to sequencing. It also enables accurate pooling of samples if this is part of your workflow. These UHR samples were amplified to obtain final library concentrations in the range of 200 to 500 nanograms. The final QC metric is to analyze the profile of the final library. Here are a few examples of what we would consider a good and a bad library. The good library has a nice size distribution with a mode of about 300 base pairs. There are no other species present on the bioanalyzer trace other than the library itself. I have also shown 3 examples of what we would consider a not-so-great library. This first bad library contains all adapter dimer with very minimal library. Adapter dimers are the result of 2 adapters ligating together without any insert. This species runs at about 140 base pairs on a high-sensitivity bioanalyzer chip. The second bad library contains library, however, there is still a substantial amount of adapter dimer present. The third library shows a library that has been over-amplified. In library amplification reactions, primers typically deplete before DNTPs. When DNA synthesis can no longer take place because of primer depletion, you have subsequent rounds of DNA denaturation and annealing, followed by imperfect annealing to non-complementary partners by way of adapter sequences. This presumably results in the formation of long, mostly single-stranded daisy chains. Because these species do contain that single-stranded component, they will retard in the gel matrix and reveals a larger secondary peak. Here are the final libraries that we generated for the UHR sample. As you can see, there are no adapter dimer or residual adapters present, nor is indication of overamplification. And therefore, we consider these libraries acceptable for sequencing. In the next part of the talk, I'll talk about the development and validation of an solution on the PerkinElmer Cyclone GFX Workstation. The Cyclone GFX Workstation offers a standard deck layout. It offers a flexible throughput, allowing for 8 to 96 samples to be run at once. There are 4 temperature locations, 3 of which allow for only temperature changes, 1 of which also has the ability to shake during incubations. Lastly, the sunken deck allows for tip storage of 26 tip boxes, minimizing user intervention. The Kappa-stranded mRNA-seq workflow on the Cyclone is broken up into 3 separate applications. This is useful because the user can then have more flexibility in regards to stopping and starting the protocol when it's convenient for their workflow. The first application includes the mRNA capture through cDNA synthesis. This application will typically take about 4 hours to run. The mRNA fragmentation and the first strand cDNA synthesis are performed off-deck on a thermocycler. At this point, the protocol can then be stopped and completed on the next day. The protocol also allows the user even more flexibility to start at fragmentation if they don't want to perform poly-A capture for the workflow. The second application includes library preparation with library amplification, again performed off-deck on a thermocycler. This application will take about 3 hours to complete, of course depending on how many samples you're running at once. The final application is a 1x ampere cleanup post-library amplification. This is conveniently also a separate application, which allows you to separate pre- and post-amp material on separate instruments if desired. Next, we will talk about the validation data that we generated in collaboration with Dr. Thomas Westerling. We assessed his samples for input titrations, reproducibility, and also compared the results to their current workflow. During the evaluation, we assessed MCF7 breast cancer cell lines, both for input titrations and reproducibility. We took QC samples at the spots discussed earlier that were indicative of library performance. Sequencing was then performed on a NextSeq 500. Post-fragmentation, we analyzed expression levels of 28S and GAPDH. Again, we observed the expected differences between the CQ score of total RNA and captured mRNA with 28S, again indicating a decrease in residual rRNA post-capture. We also examined similar CQ scores with GAPDH in total RNA and captured RNA. These results were consistent from 10 nanograms to 500 nanograms. In addition, replicate libraries were made for each input, and results were consistent between replicates. We also examined the % conversion to adaptor-ligated molecules for the MCF7 cell lines. Again, we are observing successful conversion rates with ranging from 5 to 15%. We also examined yields post-amplification using the library quantification kit. Because we had a large number of samples, we split the cycle number into two. Inputs that were greater than or equal to 500 nanograms were cycled for 10 cycles, whereas inputs less than 500 nanograms were cycled for 16 cycles. As a result, the 125 nanogram input was overamplified and could be fine-tuned in future studies. As mentioned, it is important to limit the amount of amplification cycles to the amount of material you actually need to move forward with your workflow. Overamplification results in artifacts such as amplification bias, PCR duplicates, chimeric library inserts, and also nucleotide substitutions. Lastly, the libraries were analyzed electrophoretically. Again, the libraries give a nice size distribution, and there is no indication of residual adapter dimer or unligated adapter present in the libraries. Now we're going to get into the various sequencing results that we obtained with the MCF7 cell lines. Here we assess the input titrations and their replicates for general sequencing metrics. Shown here are both mapped reads and uniquely mapped reads, which are defined as reads which are not PCR duplicates. The mapped reads were fairly consistent across inputs and replicates, and as expected, the amount of uniquely mapped reads will increase as you increase your input amount, with 500 nanograms having about 85% of uniquely mapped reads. As a result, the duplication rates upon our increase in input will also decrease. We also examined the number of genes and transcripts, which was found to be consistent across inputs. The lower figure shows the strandedness. And again, this is maintained within the 95 to 75% range. As an additional QC metric, we analyzed the percentage of exons and 5' and 3' UTR regions. Again, these values were consistent between inputs and across replicates. The remaining 30% of reads consists of reads that are unmapped and reads that map to introns and intergenic regions, which is consistent with our expectations. We also examined IGV plots from a housekeeping gene. In this case, we looked at the ACB locus across inputs and replicates. Just to familiarize everybody with these plots, the top of the figure shows the chromosomal region of interest, and a GC content trace is also included at the bottom of the figure. Each one of these lines is a specific read during sequencing, and the coverage of these regions is plotted above. And as you can see, the coverage between inputs is very similar, as is the coverage between replicates. To also compare replicates, we examined the scatter plots of the RRPKM values. To explain further, one of these dots is indicative of an individual transcript, The expression levels are plotted in RPKM values, which are reads per kilobase per million reads mapped. What this means is expression levels are normalized for transcript length and expressed as a ratio of all mapped reads. This allows for direct comparison between transcripts of different lengths as well as between samples and various treatments. And as you can see, the correlation value for both inputs exhibits a high level of correlation with R-squared values greater than 0.99, again pointing to the consistency of the workflow. We also compared the Kappa-stranded mRNA-seq kit to the customer's existing non-stranded workflow. The top trace is for Kapa, which is shown in green, while the competitor is shown in orange. Shown on the left are two examples of housekeeping genes, GAPDH and ACTB. For these two highly expressed genes, Kapa and the competitor perform equivalently. However, when looking at GC-rich regions, such as DVL1, Kapa sees an advantage. Here on the right side of the figure is a specific region where Kapa outperforms the competitor. And similarly for the low abundance transcript, GLTP-D1, again, Kapa outperforms the competitor. These features point to the efficiency of Kappa hi-fi DNA polymerase and the overall formulation of the library preparation kit itself. To bring it all back to some biological relevance, I just wanted to show some expression data. This is showing a PCA plot. Essentially what PCA tries to generate are a set of axes that explain the total variance within a data set. The first component, PC1, will explain the most variance within the data, followed by the second component, PC2, and so on. The data is then transformed and plotted in terms of these new axes and components. Points that are close to each other have a more similar variance profile than points that are further away from each other. Therefore, on the plot on the left, you can see that all UHR samples, which are shown in blue, have a similar variance profile to each other. This is very different to the variance profile of the other samples, MCF7 cell lines, which are shown in red, and the drug-resistant cell lines that we examined, which are shown in yellow. Further, replicates and inputs are also included in this assay, again pointing that they all perform similarly in regards to expression data. So in summary, what we have done is developed and validated a set of QC metrics for this Kappa-stranded mRNA-seq kit that assesses library quality prior to sequencing. We also developed and validated an automation script on the Cyclone. Along with Dr. Tom Westerling, we successfully generated libraries from 10 nanograms to 2 micrograms of input RNA on the Cyclone. General sequencing metrics and coverage plots indicate minimal differences between inputs and replicates. Kappa offers an increased coverage of GC-rich regions and more even coverage of low abundance transcripts, which offers an edge over the industry-leading kit. And with that, I would like to thank specifically Dr. Tom Westerling, who has allowed us to move forward with his samples and present this webinar. And with that, I would be happy to take any questions.
Thank you, Dr. Geldart, for that excellent presentation. We do have some questions here from the audience, so I'll start with the first one. The question here is, can you please go over again how overamplification affects library construction?
Yes. So it is important to minimize overamplification again, as this will minimize the amount of artifacts that you see. So if you do overamplify a library, as I've shown, what happens is that you get, you run out of primers, you get subsequent DNA denaturation and annealing, which allows for these large heteroduplexes that have some single-stranded components called daisy chains. If you run these on a bioanalyzer trace, what you're going to see is that these partially single-stranded molecules will run slower on the bioanalyzer trace, which gives you that second large secondary peak. These molecules will not be quantified accurately by the bioanalyzer, or qubit or nanodrop for that matter. So the most important thing to do if you do have overamplification is to quantify them appropriately on a qPCR instrument with our Kapa library quantification kit. Because qPCR does involve a denaturation step, these molecules, which are properly adapted and legit library molecules, will become denatured, allowing for accurate quantification.
Okay, great. A follow-up to this question is, if you see your library is overamplified, what can be done in that scenario?
So if you do see that your library is overamplified, there is nothing that you can do to remove that overamplified portion. They're actually, it's just an artifact and of subsequent denaturation and reannealing. So you would definitely need to quantify it appropriately with the quantification kit. And moving forward, it would be a good indication to optimize your library amplification reactions. And what we generally suggest is that post-ligation, if you're able, to take some of your post-ligated material, quantify it with the library quantification kit, and accurately determine how many adapter ligated molecules you have. And from there, you can actually determine how many cycles you'll need to obtain enough material for whatever downstream workflow you're performing, whether you're going into capture or whether you're going into sequencing. And this will kind of eliminate the chances of overamplifying.
Okay, great. Another question related to this, since it's a major QC step, is for this qPCR QC method, what's the expected delta CP for the 28S between the total and captured RNA?
Yep. So this is referring to the one-step QRT PCR kit. Generally for UHR, for example, we see a difference in CQ scores between 8 to 12. This will obviously differ depending on what type of RNA you're looking at. But for the UHR samples that we generally see, it is about 8 to 12 cycles. For GAPDH, we see, a difference of 1 cycle on average, 1 to 2 cycles.
Okay, great. Now a couple of questions about the kit as well. Can this kit be used for FFPE samples?
So if you were to use this kit for FFPE samples, I would suggest actually just using the RNA-seq. We have a Kapa RNA-seq library preparation kit. And this way you can use a ribosomal upfront module prior to the kit. Because FFPE is degraded, you won't have the poly-A tail in most cases, which would be, you wouldn't capture your material efficiently.
Okay. And somewhat similar along those lines, can this kit be used for small RNA sequencing?
No. Unfortunately, our kits cannot be used for small RNA sequencing.
Okay. So now I have a question about using quantification after ligation. So can you use quantification after the ligation step to determine the best number of cycles to perform during amplification?
Yes, you absolutely can. We actually have a calculator. If you're interested to reach out to support at kapabiosystems.com, that can actually help you with this calculation. So what you can do is you can determine, again, the amount of adapter ligated molecules, and from there determine how many cycles you need to obtain a specific amount of material.
Okay. And then we have a question here about some of the data that you presented. So in some of the graphs, the percentage strandedness was lower than 99%. Mm-hmm. So we have an attendee wondering why that was the case.
Okay, that's actually a good question. So what we think is going on is that the Kappa Hi-Fi DNA polymerase is actually very efficient and very good at amplifying GC-rich regions. So what we think is happening is that these GC-rich regions obviously don't have any A's where you can incorporate a U into the second strand. And therefore what you're seeing is that you're able to amplify bulk strands effectively for the GC-rich regions, which might show a slight decrease in strand specificity.
Thank you. And then again about the data that was shown. So what was the sequencing read depth for each input as well as the percentage duplicate?
Okay, so I can show that figure again. So here on the left, this is showing the mapped reads for each of the inputs for each replicate. Again, ranging from about 40 million to about 70 million.
Okay, great. So we have a question now about the library kit construction. So how much material do you generate for sequencing at the end of library preparation?
I'm sorry, can you say that again?
Oh, sorry. So how much material is actually generated at the end of the library construction before sequencing?
Okay. So what we did, we actually optimized the amount of cycles. We wanted to limit overamplification. So generally for these studies, we aimed for about 500 nanograms of material or less. Unfortunately, because we had such a large amount of samples, we did split them up into 2 different cycle numbers. And as a result, as I showed, we did overamplify the 125 nanogram material.
Okay. And this is the question for basically the comparison data that you showed. So what was the Q scores for the comparison data? Do you know if there are quality scores for those libraries, and how did they compare to the Kapa libraries?
So that's a good question. Unfortunately, we didn't take those specific metric points for the libraries that were generated with a competitor kit.
Okay, and then we have a question about the one-step RT-PCR kit that you mentioned. So what standards should be used with this particular type of kit?
So in that particular assay, we didn't use a standard. What we did was we just made sure that we normalized the total RNA and the captured RNA to be at the same concentration, and then that way we could directly compare against each other. What we were looking for was a change in CQ score.
We have a question about for adapter dimers with this kit. So is there a process to get rid of adapter dimers if you see them using this kit for library construction?
So the first thing I would recommend, if you do happen to see consistent adapter dimers, I would suggest optimizing your adapter concentration. Most likely, if you're seeing adapter dimer, you'd want to try to play with the adapter concentration to minimize this as much as possible. If you do go through library preparation and you see that you do have adapter dimer, the best thing to do, depending if you have enough material, is to do another cleanup step.
Okay, so this could be, is that the cleanup step? Do you recommend gel electrophoresis and then excision of that, or what was the recommended cleanup step?
I would suggest just an additional 0.8x, 1x ampere cleanup.
Okay, great. We have a couple more questions about the kit itself. So there's a question whether the kit includes controls, if there anything that's already included. If not, what would be the recommended controls?
No, the kit does not include controls. And if you wanted to evaluate the kit, again, generally what we use as a kind of a high-quality RNA sample is the universal human reference RNA from Agilent. So if you wanted to kind of evaluate the kit before moving on with, important samples, that would be my suggestion.
Okay. And we have a question about the amount of RNA that you could use to begin this library preparation. What is the minimum amount and the recommended amount?
So the kit, as it stands, is for 100 nanograms to 4 micrograms of input RNA. But as you saw, we were successful in this particular analysis, and we're able to go down to 10 nanograms. But again, that is dependent on the type of RNA that you're using, and the quality, of course.
Okay. So how many, does this kit actually allow for multiplexing in the sequencing step? Is there anything within the protocol that would limit that?
No. To multiplex, basically what you would have to do is, of course, you'd have to determine yourself, what instrument you're sequencing on, whether it's the MiSeq or the HiSeq, what kind of species you're looking at. Again, figuring out what kind of coverage you want for each sample, and kind of figuring out how much multiplexing you want to do. And then all you would really have to do is generate each of your libraries with different barcodes, so that enables you to pool effectively. And from there, after the library prep, I would strongly suggest the library quantification kit, which gives very accurate concentration information, allowing you to pool accurately.
Okay. And the final question we have at this time is if there were instructions within the protocol of this kit for the different QC steps, and if so, where can these be found?
So they are not included in the kit itself. If you would like instructions or more guidelines on how to perform these QC metrics, you can email support@kapabiosystems.com, as shown here. And we'd be happy to help you with those QC metrics. And again, this webinar will be posted online, so if you want to watch it again and get more information about the QC metrics, that's also an option as well.
Okay, great. Thank you so much, Dr. Geldart. That concludes our Q&A session, and we'd like to thank you all from Kapa Biosystems for joining us today. As Dr. Geldart mentioned, we will be posting this recording to our website, and we'll send you the link shortly. So thank you again for joining us, and have a great day.